Good morning. Today we're going to be talking about probability and Punnett squares. Okay, and we're going to be meeting tomorrow to go over this. It's just a warm up. Just look through some reminders from yesterday, just some review, like look at what genotype and phenotype is, homozygous versus heterozygous, that type of stuff. Just make sure you understand that stuff. But today we're going to be looking at specifically probability and using Punnett squares and creating Punnett squares. So probability is just the likelihood that an event's going to occur. So if I flip a coin, what's the probability it lands on heads, right? It's 50-50 every time. If I do it 100 times, the 99th and 100th time of me flipping a coin, I still have a 50-50 chance of landing on heads or tails. So the principles of probability can be used to predict the outcome of genetic crosses, all right? So a Punnett square is just a diagram that shows how genes combine for specific traits, all right? So we're looking at one trait at a time. Sometimes you can do multiple traits, but we're looking at one trait at a time today. So first, we need to know what the law of segregation is and what a monohybrid cross is. So the law of segregation just states that the separation of alleles during gamete formation. So remember, gametes, like meiosis, only have half the chromosomes. Remember, they didn't replicate. So you may get, say we're looking at the ability to roll your tongue. You may have gotten the trait that you can roll your tongue from your parents, or you might not have. Remember, when it comes together, you have two letters, like the big A, little a. When they separate for sex cells and for gamete formation, you get one of those letters at a time from your parents. Remember, there's only 23 chromosomes from mom and 23 from dad. All right, so those things separate. And a monohybrid cross is just looking at one trait at a time. And that's what we're going to look at today. So again, the law of segregation, all allele pairs separate during gamete formation, and they randomly unite when they come back to fertilize, all right? And there are four main concepts related to this principle. So a gene can exist in more than one form. So you can have the tongue roller versus non-tongue roller. Organisms inherit two alleles for each trait. You get one from mother and one from father. Plants get one from one plant and one from the other. When gametes are produced in meiosis, the allele pairs separate, leaving each cell with only a single allele. Remember, half the chromosomes. When the two alleles of a pair are different, one is dominant and one is recessive. All right? So the first Punnett square, what would be the outcome of crossing a heterozygous tongue roller, that means that's dominant, with a homozygous non-tongue roller? The ability to roll your tongue is dominant. So here's the alleles here. So you got the big A from mother, little a from father, or that's the mother, that's the father, okay? So you're going to see how these come together, right? How you would do this is like this. You sort of separate this top, the big A and little a, little a. Just coming back from this here, right? I got these from here. So just separate this out. Usually you would have a box here with four boxes within the box, but it couldn't cut and paste. It didn't cut and paste over, so sorry. But then what you do, after you separate them out like this, you come together. So you make this A and this A come together. This A and this A come together. This little A and this little A come together, and this little A and this little A come together. So they're going to form these right here. All right, that's the randomness of how they come together. So if I got a big A, little a from mother, during her meiosis, they separate out. Okay, little a from dad, they separate out. But when they come together to make a new fetus, they come together. So the outcome would be 50% or big A, little a, and 50% or little a. In this case, 50% could be tongue rollers because that's dominant, but a 50% chance that you might be a non-tongue roller, all right? So the next one, what would be the outcome of crossing a heterozygous long-haired cat? So long hair is dominant with a homozygous long-haired cat. So that's showing, so the alleles would be like this, all right? So you have this one little allele here, same thing. Separate them out and then come together from the separate angles. These two here come together. This one and this one come together. The small L and the large L come together. And the small L and the large L come together. So the outcome is 50% homozygous dominant and 50% heterozygous dominant. All right? But with those two things, 100% will be long hair. And there's a third Punnett square to practice here. Just go ahead and go through that. And then there's a review thing here that looks at Mendel and Punnett squares, just reviewing over it. Just watch the first 10 minutes or so of it. It's a longer clip, but it's a good review of Mendel and Punnett squares.